Thank you very much for the invitation to come and speak to you today. So I'm going to be talking about some of the work which we are doing at the University of Leeds. And the title of my talk is Plants on Prescription. So our research is interested in understanding how emerging contaminants, mostly pharmaceuticals, hence the prescription, enter into soil and plant systems, and then what this means for the health of ecosystems. But before I start, I just wanted to try and provide a little bit of, I guess, a background or some information about me and my research interests. And to do this, I actually took a bit of a trip down memory lane and pulled out some photographs from my research career to date. So this experiment is actually the first experiment I set up as a PhD student. So this was looking at how soil properties can affect the uptake and accumulation of contaminants into plants. And it looks small, but this involved sieving about 15 kilos of soil to set this up. So this was a huge experiment, hence the picture, because I took it for, to remember for a long time. Over here, this is, I worked as a postdoc in Australia for a couple of years, and I landed, and 24 hours later, they had me digging soil in a field to get set up for some experiments. So yes, this is me looking very keen. This is the first experiment that I set up in Leeds when I started as a PI. So this is a soil leaching column. If you can't see this already, the theme is soil. So when I was trying to think about what I do, everything seems to come back to soil at some point. And we, in Leeds, we do a mixture. So we do laboratory-based and greenhouse trials. But we've also started to do more field-based work. And this was us last summer. You wouldn't know it was summer because it was very, very wet. But this was us out in the field collecting some soil samples as part of one of our experiments. We have a number of international projects as well. So we work in the UK, but in a range of different global contexts as well. And one of the most recent trials we've got, this is in China, and it was a field scale trial that we set up to look at manipulating CO2. So we could look at how this changes the uptake and accumulation of chemicals in a changing climate. And as well as the research, we do quite a lot of outreach in our group. So this picture is actually from, I think it was the Great Yorkshire Show. And we set up a school's exhibit to try and think about soil properties and how this can influence crop growth. So just a little bit of a snapshot about some of the research that I do and really where this has all started. But back to the topic of the talk today. So really my research sort of falls under this concept of a circular economy and the reuse of resources. So hopefully you can see this and it's illustrated on the slide here. So when we're talking about the reuse of resources, we're talking about these circularity principles. So resources are things such as wastewater effluents, which can be reused as a source of irrigation. We've got here sludges from wastewater treatment works, and these can be reused and applied to land as a source of fertilizer. And then we've also got here the animal wastes, which can be repurposed on agricultural land, and you have this sort of circularity principle here. But the one thing which we're interested in is how do contaminants or emerging contaminants enter into these systems, and how does that then affect or present any potential risks to human health and to the environment. So if we take human consumption for an example, and we think about pharmaceuticals, does anyone know what happens to a medicine or a pharmaceutical after you take it? Any suggestions? Yeah, you've got the theme. So we excrete a large proportion of the medicines that we take. So typically, a small proportion of the medicine will get used to elicit the therapeutic effects in the body, and then the rest will be eliminated, or it can be metabolized in the body, and then this enters the waste stream. So this is where we're interested in trying to understand what happens to these pharmaceuticals which are inadvertently entering into these waste systems, and then through the reuse of materials, so the effluents and the solids, we're interested to understand what's happening to these chemicals. Because wastewater treatment plants are in operation and they do a great job at removing a number of contaminants, but they're not designed to remove a majority of the pharmaceuticals or emerging contaminants that we're now dealing with. So quite often they can enter through the sewage system, and then they can remain present through the treatment process and then be released in the byproducts of the wastewater treatment plants. And it's those byproducts that we're using now to add nutrients to soil and to meet our irrigation demand. So the work which we are doing in Leeds focuses, yes, broadly on emerging contaminants. So if you're not familiar with this term, this is chemicals or contaminants that are newly detected in the environment, or they're re-emerging in the environment, and we don't know very much about their potential for human health or ecosystem risk. So has anyone got any ideas of what an emerging contaminant might be? There's some clues on the slide. Has anyone heard of microplastic? Yeah. What about PFAS? Yeah. So we work primarily with pharmaceuticals of human use and veterinary origin. We also have work ongoing with microplastics and PFAS as well, and some pesticide research as well. And when I'm talking about this reuse of resource and the use of human waste, I think this is really important because in a number of agricultural contexts, we are it's being framed at the moment that the reuse of this waste is a future solution to meet our demands for nutrients and water. But what I would like to argue is that it's not necessarily a new concept or a new technique. This is something which we have been doing as a society for many, many years. So I was reading this paper the other day and I thought it would be really relevant to this talk. So this is showing water and wastewater conveyance facilities from prehistoric times. And in here on the left-hand side, you've got these conveyance facilities that transport stored water into farmland. And here we've got storage tanks, which were then used for water for agricultural irrigation purposes. So the concept that we're talking about here, so the collection of waste and then the reuse of this, is not something new. It's something that we've been doing for quite a long time. But what we're dealing now with as a society is a very different picture, in that we have different, we are a very consumptive society, let's put it that way. So we have high use of chemical products. We rely on medicines. We rely on personal care products. We have a very high consumption of these chemicals. We've changed our approaches and our sewage. We have large centralized collection facilities where waste is all coming together from different sources. So we've got industrial waste, we've got domestic waste, we've got hospital waste, and it's coming together and you've got this added layer of chemical complexity on top. And so what we're dealing with is this concept of reuse, but in a very different landscape, because the actual the makeup or the components of this human waste are very different to what was being used in those earlier examples. And the work which I'm going to talk about now for the remainder of this talk is really centered on pharmaceuticals. And this is because these are one of my key interests as an emerging contaminant, because they're biologically active. So when I talked about the process that occurs following their use, so if these chemicals are not fully metabolized or they're not fully transformed in the body, then that biological potency, the thing that you're actually taking the medicine for, remains. And then there's the potential for that to enter the environment, interact in the environment with different receptors. So I think pharmaceuticals are a really unique, but a really important contaminant that we need to better understand, because if they have that conserved biological potency, and that's what I'm going to try and introduce and talk to you about today. But quite often when you talk about pharmaceuticals, the first thing on everybody's mind is wastewater or river pollution or waters. So if anyone seen any of these articles in the news recently about combined sewer overflows being discharged into rivers, and yeah. So this is quite often when I talk about pharmaceuticals in the environment, most people associate the pollution with waters. So you've got your rivers, your lakes, your streams. And what we're interested in is we do have projects on water, I should add. But what we're interested in is the reuse of wastewater as a source of irrigation. So this is a trial which we had in Israel, and it was set up to look at wastewater irrigation on cropland. This is biosolids on one of our field sites in the UK. And this is a pile of biosolids from a wastewater treatment plant before it was then spread onto land. And then this is an example of manure spreading on a field as well. So we're interested in understanding how pharmaceuticals from these different pathways can enter the environment. So it's not just wastewater going into rivers. And when I try and break down what the research is that we're interested in our lab at Leeds, I've tried to sort of conceptualize it in these three different themes. So firstly, we're interested in the pathways. So by pathways, I mean, how do these chemicals make their way from use into the environment? So what are the different mechanisms? What are the different processes by which these contaminants can enter and become present in the environment? Now, if we've established that they're present in the environment, the next question that we're interested in is their fate. And so the fate means things such as, do they disappear? Do they transform? Do they get taken up by biota? So what happens to the chemicals when they're present in the environment? And then ultimately, I think I'm standing in the middle, but what we're trying to do is pull that information all together to try and understand what the risk is. So what's the risk to human health and what is the risk to ecosystems? So that's really where we're trying to drive this research, is to try and increase this understanding and to pull this all together to better understand what the risk is. Because unless we know what the risk is, we can't mitigate and we can't put measures in place to enable us to do this safely. So this is really where I started at Leeds. So I started in Leeds in 2018. And for those of you who are not familiar, Leeds is a commercial pig farm. And so my initial reaction was like, we need to look at this in a bit more detail because pigs produce waste. Waste goes onto land. Nobody's looked at this. This seems like an obvious thing to me. But yes, it took quite a while to get this study to completion. But this is one of my PhD students' papers. And we studied the farm system. So we took slurry samples. So these are in the slurry tanks. We took samples following the application of the slurry to land. And we did this over the course of a year. And the university farm also had a series of boreholes. So I didn't set these up. This was very lucky because it then enabled us to look at the groundwater pollution as well. So we've got this sort of somewhat complete system to be able to look at where the different antibiotics are going. So the chemicals that we are interested in were antibiotics listed here. And these were, we targeted our analysis towards these because they're used on the farm. So they're used in the pig production. And I appreciate these numbers are quite small, but what I'm trying to show here is that we were able to detect antibiotics in the slurry, in the soil, and in the groundwater. We were able to detect the quite high concentrations in the slurry tanks. And if you can see, then you've got a factor of 10 to 50 times lower in the soil. So we're still able to detect these antibiotics in the soil. It's just at a lower concentration than what's in the slurry. And to be honest, the most surprising thing for me was that we were also able to detect them in the groundwater. So we were able to detect antibiotics even down to 47 meters in the groundwater. And what we were able to see is an increasing concentration in the groundwater following the application of the slurry to land. So over the experiment, or over the year of monitoring, we could see this increase in the concentration. So this study in the farm really started our interest in the monitoring and trying to better understand the pollution in our local environment. So a lot of the work which I've done in the past when we're trying to look at the concentrations of these chemicals in soil has largely focused on study sites in the US, in Australia, and in Israel, as I said before. But we don't have that level of detail in the UK. So we devised this spade monitoring campaign to try and better understand about the presence of emerging contaminants across soils at a more local level. So we were interested in soils that had biosolids applied, manure, digestate, and we tried to characterize these soils to better understand the presence of contaminants on home soil. So a lot of the monitoring that has taken place in the UK to date has largely focused on what we would call traditional pollutants. So your things like heavy metals. But what we were specifically focused on were these organic chemicals, or these emerging contaminants that I introduced in the beginning. So we did a combination of different approaches. So we actually went to several farms that we had identified and we had working relationships with, and we did a comprehensive sampling campaign. So we took soils from multiple different fields. We had different sampling approaches. We took samples from different depths, and we had quite a good coverage in this respect. But we also tried to get extra samples as well. So we invited farmers to send in their soil samples by post. So we were also able to analyse these, but they just didn't have as much sort of depth to the sampling campaign that we had from some of the other sites. But in total, we had over 400 samples to analyse. And we analysed these in our lab, and we took a sort of two-pronged analysis approach. So we developed a list of target chemicals that we thought would be present. So this is based on our work from other study sites, so in the other countries that I mentioned. So we picked up on a few chemicals that seem to be quite persistent and seem to be present in multiple different time periods when we were sampling. And we also had knowledge about what was used in the UK, the high consumption chemicals. So we devised a list of, I think it was 18 chemicals in total, and we did a targeted analysis campaign. So this is basically the method allowed us to determine the presence, but also to quantify the concentration of the chemical. So this was quite an in-depth bit of method development, but we could get a really good picture on what was present and the concentration. But then we also coupled this with non-target screening. So this enabled us to create a chemical fingerprint of what was in the sample, but we couldn't determine the concentration. So that's why we've got these sort of two complementary approaches. We had one more in-depth type of analysis, and then we have this other sort of chemical fingerprint. Let's look whether it's present to try and create that bigger picture of what might be present in the soils. Now, this is a very busy figure, so I appreciate I'm not expecting you to read it all. But what I really wanted to include this for was just to show you the diversity of what we were able to find. So this is from the targeted analysis. So this is where we had that predefined list. So we were looking specifically for these chemicals. And what you can see here is we have a range of different chemicals that we're able to detect. So some ones I want to highlight here, we've got carbamazepine and lamotrigine. So these are anti-epileptic pharmaceuticals. So these are human use chemicals, but these were detected in agricultural soils. We also have, so some of the higher concentrations were what you would typically expect. So these are veterinary antibiotics and some pesticides. But yeah, we have detectable concentrations. Diclofenac is an anti-inflammatory that you can get over the counter. And we've got concentrations here ranging from 0.001 to 100 microgram per kilogram in soils. So what was really interesting in our results is that you really needed to have a good relationship with the farmer, and you needed to have some site history. Because when we analysed this data for the first time, we had labelled that this was a biocide or a sludge amended field. But we were finding lots of veterinary medicines, and we were like, this doesn't make sense. But then when you actually understand the site history and you learn about how the farm is managed, that's when this sort of the bigger picture unravels. And you can see that people don't just apply year on year the same material to land. So some farmers applied manure for a couple of years, then had a rest period, and then applied some biosolids, and also vice versa. So it's a very complex environment. It's not easy to predict what's going to be there without that understanding of how the land and how the farm has been managed in the past. And we also took what we thought were control samples, because we're scientists, we wanted a control to compare to understand that there was no contamination. And in one farm, we were taking this sample and we took hedgerow samples, the soil from just beneath the hedgerow, because we thought this would be it's similar soil properties to the main field, but it hasn't had the direct application of the amendments or the manure or the biosolid. And the farmer came over and was like, "What are you doing? This yeah." And I was like, "I'm taking my control sample." And he was like, "Well, that's not a control, because if we've got any manure that's left over, we just chuck it on the hedgerow." He was like, "Great, there goes my control." But I think the point I want to make is that having a relationship and trying to actually understand what was happening and what the management practices were was really important for this work. So these are some of the pharmaceuticals just to pull out. So this is the results from the non-target screening. So this is the chemical fingerprint. So we can tell whether it's present or absent, but not the concentration. And we were able to see quite a lot of plasticizers. We saw research chemicals. We saw dyes from materials and textile manufacturers. Pharmaceutical research chemicals. But probably you're not surprised to hear now, we also found a lot of human and veterinary use pharmaceuticals. So this is just a bit of a heat map to show what we found. But the most commonly detected pharmaceuticals were antibiotics. But then we found quite a lot of antifungals, antidepressants, non-steroidal anti-inflammatories. So we've got quite a range of different contaminants that we were able to detect in soil. So this is really trying to provide the foundation and show you that in the UK we've got this background or this contamination coming from the reuse of resources. So this leads really nicely on to the question of, so what happens? That just because it's introduced, what does that mean? So where do these chemicals go? And this is a slide that I start some of my my talks for the students with, where it's trying to think conceptually about what's happening to these chemicals once they enter soils. So I think most of these processes are going to be familiar with you, but just in terms of pharmaceuticals, so we're really interested in mobility. So absorption and desorption to how well the chemical sort of sticks or can be lost from the soil is really important. Because if it's stuck too tightly to the soil, it's not going to go anywhere. So its behaviour is very different to a pharmaceutical that's more sort of mobile and can move a lot in the soil. So if it's mobile, for example, it could run off into a stream or a nearby water body. It could leach into the groundwater, as we saw at the farm example in one of the earlier slides. But what we also need to think about is the potential for any removal or any loss. So there's environmental processes like photodegradation, microbial degradation, which can break these chemicals down so that you would see a reduction in their presence as well. But this sort of leads on to, so this is one of the first studies I ever did. And this is trying to understand what happens to a pharmaceutical in a soil plant system. So what we were really interested in is understanding how different pharmaceuticals with different properties, how they behave and where do they go. So what we were able to show is that the hydrophobicity, or effectively how well the chemical sticks to something, is really important when thinking about the fate of pharmaceuticals in soil plant systems. So absorption, so how well it sticks to the soil, and degradation are very important. If the chemical is not in the soil solution or in the pore water, it's very unlikely to be taken up into the plant. If it's really hydrophobic, then it's predominantly going to stick to the roots. Whereas if it's sort of moderately hydrophobic, then we see the chemicals able to cross the root membrane and it goes up into the shoots. So where the sort of chemistry of the chemical is really important to understand what happens to it in this system. And then I followed this up with another study when I was a postdoc. So I was really interested to know what happens to these chemicals. I mentioned it before about sort of the transformation or the metabolism of a chemical, so how it can change in the body. I was really interested to understand whether that happens in the environment and in plants. Because quite a lot of the research to this point had just been focused on what we call the parent compound, so the active chemical that you're prescribed if you go to the GP, for example. That is where a lot of the research had focused, but I was really interested to try and understand what happens. Does that parent chemical stay around, or does it transform and change into something else? So we worked with a set of drugs called benzodiazepines. So these are anti-anxiety drugs. And one of the most commonly used anti-anxieties is diazepam. So I carried out the study where I, for want of a better, I guess, sort of phrasing, you basically get the drug, you can crush it up and you can stick it into the soil. So you spike the soil directly with these anti-anxiety drugs. And then you look at what happens, where does it go? And we started the experiment with, yes, diazepam. So this is the parent compound, added to the soil, and we used radish as a model crop in this experiment. And then at the end, what I was able to see was the presence of diazepam, so the parent compound, but also these metabolites. So nordiazepam, temazepam, and oxazepam are all anti-anxiety drugs that are prescribed in their own right. And what we were able to see was this in-plant metabolism processes were converting this diazepam into these metabolites in the plant. So we couldn't see them in the soil, but we could detect them in the plant. And I think this is really important because when we're thinking about chemical risk, we are very focused on the parent compound, but these are all biologically active chemicals in their own right. So we can't just think about the parent compound, we need to think about this bigger picture. And yes, what we're able to show is that the transformation of the diazepam followed the same process as the transformation in the human body. So you've got this sort of very quick formation of diazepam to nordiazepam, and then you get nordiazepam converting into oxazepam, and that's the final metabolite product. And we could see that present in the plants. So far, what I've hopefully tried to present to you is we've got the case of the different pathways introducing these pharmaceutical chemicals into soils. I've then talked about the fate. So what happens to these chemicals when they enter the soils? How do they behave in these soil plant systems? And so the question at this point I normally get asked is, so what does this mean? What are we talking about here? So you've shown that there's presence, but what's the deal effectively? So the questions and sort of what we know so far are on this slide. So what we've been able to show is that there's a presence of these chemicals in soil, and that environmentally relevant concentration, so what we're seeing in the environment today, they can be taken up into crops. We've been able to show, so we've got some studies that have shown that antibiotics in particular can impact on soil microbial community composition. And this is not a massive active area of research for me, but I do have collaborators, and they are looking at the impacts on human health. I'd argue that this is something that's largely unknown, but we have been able to show that crops that are irrigated with wastewater containing pharmaceuticals are consumed by humans, and then we can detect the pharmaceuticals in the urine following the consumption. As for what that means, I think there's a lot more work that needs to be done here. But what I really set about in Leeds trying to understand is potential impacts on the crops. And that's what I'm going to finish the talk with. So I'm going to, first of all, introduce some of this work I started with. So when there was this emerging evidence that these chemicals were detected and being able to be taken up into plants, everyone said, let's do a standard ecotox test. Let's do a seedling germination. Let's do something to understand whether there's a risk or not. And quite a lot of the studies were coming back that the sort of point of lethality or the impact on the crop was at very, very high concentrations. So not the concentrations that we would be seeing naturally in the environment. And this is probably, well evidenced here. So this is a study that my master's student did, and it was working with the zucchini crop in Australia. So this is something that is widely grown under market gardening sort of processes in Australia, especially in South Australia. And they use wastewater to irrigate these zucchini crops. So this is why we use zucchini. But what you can see here is this sort of following pollination, you get these zucchini fruits and they shrivel up and die. And this was in response to an anti-epileptic pharmaceutical, carbamazepine. So this is what we've detected in soils in the UK. So the plants, they would look pretty healthy for a while, and then it was about between 15 to 20 days, they would then end up like this. But the key point here is the concentration. So this was happening at concentrations that I would say are not environmentally relevant. So 14 milligrams per kilogram is about 50 to 70 times higher than what we're seeing in the soils in the UK from our monitoring campaign. Okay, so we're seeing this effect, but it's very high concentrations. We also did this study here. It was actually with zucchini again. And what you can see, this is increasing concentration of carbamazepine, the anti-epileptic pharmaceutical. And this is 10 milligrams per kilogram. And you had this sort of dead spots on the leaves and burnt leaf edges. But these are environmentally relevant concentrations here in B and C, and it's not as sort of evident in terms of the impact. But I think rather than leaving it there, this is when I started to ask more questions, and then this has opened up a whole new area of research. So I'm going to just talk about 2 examples now, and that's where I'm going to finish the talk for today. But this really, for me, sort of created many questions in my mind. So at the really high concentrations, we can see these sort of very toxic effects on the plant. But I was then interested, so what's happening at the lower concentrations? What's happening at the environmentally relevant concentration? Just because there's not a visual or sort of lethal toxic effect on the plant, does that mean that nothing's happening, or is something else happening in these systems? So this is really where the whole area of sublethal effects that I started at Leeds really came from. And one of the studies I just wanted to introduce is one that was published quite recently, but it was in collaboration with Katie Field at the University of Sheffield, and it was looking at the impacts of antifungal pharmaceuticals on plant-fungal relationships and whether this could impact on nutrient acquisition and accumulation. So we were really interested in antifungals because we were able to detect them in soils. We had reported environmental concentrations, and the whole point of taking an antifungal as a human is to destroy the fungi. So I was like, what's happening to plant fungal systems? Like there's something there to explore, right? So this is where, yes, I started working with Katie on this. So I'm happy to share the paper if you'd like to know more about the methods, but I'll, yes, briefly introduce it for the topic, well, for the purpose of this talk. But we used radio and stable isotope tracing techniques for this experiment. And we grew different plants from seed, and the antifungals that we used were ones that we've detected in soils and are quite commonly used in the UK. So we used clotrimazole, miconazole nitrate, and fluconazole. So we did this experiment with wheat, with spring onion, and with lettuce. So these crops were grown in this arbuscular mycorrhizal inoculated soil with and without the presence of the antifungals, okay? And we used isotope tracers to quantify the nutrient exchange and the carbon flow. We analyzed the plant tissues for the isotope uptake and for the biomass. And then we also did some root and some soil DNA sequencing as well to try and understand and create a better picture as to what was happening. But really, what we were trying to look at was the impact of the presence of antifungals on this carbon for nutrient exchange in these systems. So the first experiment we did, so this was on wheat, and what we were able to see is that phosphorus transfer was significantly reduced in the presence of these antifungals. But what was really interesting is that we actually saw no impact on the degree of mycorrhizal colonization. So the colonization of the roots wasn't affected, but the phosphorus transfer was. So we repeated this experiment, this time with spring onion. And again, we saw a reduction in phosphorus transfer, but this time we did see an effect on the colonization of the roots. So we saw a reduction. So the colonization by the fungus on the roots was reduced by about 80% in the spring onion. Yeah, so this is just the figure for the reduction in the colonization. So we've got here this is the results from when we had the antifungals present, and then this is the control, and the same here. So this is actually for the phosphorus. So we were able to pull all of this data together. So as I said, we've got the data for lettuce, we've got spring onion, and we've got wheat as well. So what we were able to observe was this sort of broad response across phosphorus. So across multiple different species, we saw this reduction in the acquisition and the transfer of phosphorus into these plants. When we looked at the nitrogen results, we didn't see the same effect. So it was only phosphorus that was affected, not the nitrogen. So we did, as I said, we did some DNA sequencing, and we were able to show that there was a decline in fungal diversity following this analysis. And the sort of conclusion that we came to at the end, so not only did we see reduction in colonization for the spring onion and the lettuce, we saw reduced hyphal length as well for the fungi. And so the sort of working hypothesis that we came up with was that we had this diminished capacity of the fungi to access and to solubilize all the sort of phosphorus pools. And so you had this reduced capacity of the fungus to then transport the phosphorus to the plant. And we said that this was only relevant for phosphorus because of, and this was linked to the mobility of phosphorus, which was low in comparison to nitrogen. So nitrogen, with the ammonia and the nitrate being more mobile, we weren't as reliant on this fungi network to try and collect the nutrients. So the last study, which I'm just going to briefly introduce because I'm cautious of time, is a piece of research we've got ongoing at the moment in Leeds, and it's looking at more longer term and molecular effects, again using radish. And what we're really interested here is do pharmaceuticals have this longer term impact? And so what we've done is we've carried out a study where we've taken radish to seed, we've collected the seeds, and then we've planted and grown them again. But during this experiment, we've exposed some plants to diclofenac, which is an anti-inflammatory drug, some plants to a female sex hormone, so 17-beta estradiol. And what we saw, this was actually one of the biggest investments in time and money for an experiment I have ever done. And when we got to the end, so it was probably about 6 months from the start. So normally a lot of our ecotox work is maybe 12, 20 weeks maximum. So this was a much longer trial and took a lot of planning to do. And we got to just before the flowering stage, and I was like, nothing's happened. Like, there's no response. And I was like, this is great. This is still a result, but this is a very expensive no result experiment. And I started to sort of plan the next piece of work, I guess. So we saw no significant difference on the biomass for any of the plant organs that we harvested. So no significant difference to the controls for the diclofenac or the estradiol treatment. The biomass was not affected. We saw no effects on the photosynthetic activity for these plants. And so I was like, okay, it's fine. But then what we were able to see, we did keep the experiment running. We followed our plan, and we saw a significant effect on the number of seeds, or the total seeds produced under these treatments. So here, this is the table. So the control plants produced a total of 1.5 grams of dry weight seed. We've got a reduction to just over 1 gram in the 17-beta estradiol treatment. And then in the diclofenac treatment, we've got 1/10 of the seeds that were produced in this control. And at this stage as well, we also saw flowering was delayed in these treated plants as well. So in the estradiol treatments, flowering was delayed by 12 days in comparison to the control. In the diclofenac treatment, flowering was delayed by just over 20 days in comparison to the control. And we also had delays on the formation of the seed pods as well. So, as I said, we grew the plant from seed, we exposed them to the different pharmaceuticals, but then we collected the seed. And then we've just finished this sort of second phase where we took, again, the plant all the way to seed. And this is just to sort of show this sort of, this is something we're working on at the moment, but this sort of memory or this hereditary effect. So here, this is the total seed yield from the first phase of the experiment. So this is the control, this is the diclofenac treatment, and this is the estradiol. But these are the seeds that were collected and then planted again, but not exposed to any chemical. So not a repeat exposure of the chemicals in the second phase. And you can see that again in the diclofenac and the estradiol treatment, you've got this reduction in total seed yield when there's been no additional exposure to the chemicals. Yes. So this experiment here, so it was a controlled growth trial. So we set it up in a growth chamber. We irrigated using synthetic wastewater, but at concentrations that we're detecting in the environment, and following a standard irrigation procedure that you would do. That was an important point. I should have said it. So yes, this is really where I wanted to sort of finish and bring the work to a close, because something else we've been trying to work on. So a lot of the work I've talked about is experimental, lab or field-based, but we've also done quite a lot of modelling to sort of extrapolate. Because if we think about the environment and the world we're in, there's, I think, 300 and what was the latest stat? 350,000 different chemicals approved for use. I don't need to tell you how many plants there are. This is a huge combination of different potential scenarios, and we can't experimentally work on everything. So what we've been trying to do is sort of use some models and develop some frameworks to better understand where we should prioritize our efforts. So this is something that I think it was 2 years ago when I was looking back trying to think about some of the work we've done, and I was like, are we actually working on the right things? Are we working on the right pharmaceuticals, and are we working on the right plants? Because a lot of our methods in our lab, so our chemical methods, are driven by the availability of analytical standards. They're driven by how well the methods work on an instrument or a bit of kit. And it seems like a biased approach to be thinking about research in a way that's driven by the availability of the chemistry. So we tried to think about this in a different way. So we developed a framework to predict the accumulation of pharmaceuticals into crops, to take an unbiased view. So we took chemical structure alone at the beginning here, and then predicted its consumption, its fate in wastewater treatment, its fate in the effluent sort of prior to irrigation scenarios. We've got fate in soil, and then uptake into plants. So there's been a huge effort to try and build this framework. But what we've been able to do is prioritize and highlight where understudied chemicals are. And this, I think, it's set out with, I want to work out what chemicals we need to be working on and what are the important things we're missing. But I am still shocked by the results that we were able to produce, in that we are not even scratching the surface of which are the most important combinations and the types of chemicals we need to work with. So through this prioritization and through this modelling, a couple of key take homes. So presence in wastewater does not necessarily mean that it's going to be taken up in a plant or there's going to be reported presence in a plant. So it's not a very good predictor. But I think the most important statistic is that for the top 30 chemicals that we predicted to accumulate in plants, we have no data in relation to their presence in soil or uptake into crops for 53% of them. So over half of the top 30 chemicals we think are predicted to accumulate to the highest concentrations, we have no data or no understanding of at least half of them. So all of the chemicals which I've shown you and all of the methods which we've developed, we really now need to pivot and actually think about these underexplored chemicals to really understand what the risk is. So I'll highlight some of the ongoing work. So this, yes, I appreciate I've taken your time, but this is only a sort of a snapshot of some of the work we're doing. So there's a lot more which I'm happy to talk to you about. And yes, you'll have these slides. So just to have a look if there's anything that you would like to explore or discuss further. But yes, I think that leaves me to really acknowledge the fact that this work is a combination of, it's a lot of years of work put together, but it's really driven by people other than me. So yes, to acknowledge my research group and collaborators. Thank you very much.